Okay, I think we're going to start the session, and wanted to thank you for attending this session on the last day, early in the morning. I guess if you're here, you're really very interested in the topic, and that's wonderful to have this session. So today we're going to speak about the technology roadmaps for achievable manufacturing. And what we'd like to do is we want to illustrate the fact that successful implementation and commercialization of cell therapies relies on the development of consistent and robust manufacturing platforms, which is easier said than done, as we know, specifically as we are dealing with living drugs and predominantly at this time with personalized cell therapies, although obviously many of us are working also on broadening these source of material. These cell therapies pose unique challenges in terms of the manufacturing, the standardization, and distribution. And in order to establish robust and reproducible manufacturing processes, it's advisable to follow quality by design principles. The goal is to really identify quantifiable molecular and cellular characteristics that will ensure product safety, efficacy, and potency, and that will hopefully drive the manufacturing process from the starting material to the final product. One of the goals also is to obviously try to develop more automated platforms with online analyticals, which will help monitoring the processes over time and ensuring the success of the processes. So our three distinguished speakers today will address some of these issues in the context of their own work and interests. And without further delay, I would like to ask Krish Roy from the Georgia Institute of Technology, who will discuss achieving large-scale, cost-effective, reproducible manufacturing, a roadmap to 2025. And please.
Thanks, Isabel, and the organizers for inviting me to talk here. It's great to be part of ASGCD. I remember when the C was not there. It was just ASGT. As a grad student, I started coming to, and then the C got added, and luckily it got added because that's what I was going to talk about today. And so the talk that I'm prepared is sort of more of a vision talk looking forward the next 10 years. There is not my own data here, but I'm going to present, Isabel asked me to talk about a roadmap that we worked together very closely with industry and NIST and NIH and FDA, and to present a lot of the findings of that. And then talk a little bit what we are doing at Georgia Tech. So obviously, this is preaching to the choir. I don't need to talk about the incredible advances that you all have made in the cell therapy space, not only on immunotherapies, but a huge array of stem cell therapies. This is sort of the latest out of CNN on the autism trial that's going on at Duke with John Kurzberg's group. Obviously, this has been Science's Breakthrough of the Year, Time Magazine. So there is incredible amount of activity and industry interest in this space. I like to show this slide a lot of the time because we kind of forget about this. We as a taxpayer pay about $3.2 trillion in healthcare costs, trillion with a T, each year in the United States alone. And the majority of that actually do not come from prescription drugs, despite what the popular media tries to tell us. It actually, more than 60% of it comes from long-term patient care and for chronic diseases. And cell therapies and gene therapies are a means for alleviating a lot of those. Even we can make a 10, 20, 30% dent in that cost. That's a tremendous economic benefit to the nation. And not only the direct cost of all of these, but the indirect cost of lost productivity and other associated cost ranges to almost more than $1 trillion as well. So from an economic point of view, it makes absolute sense to have tremendous investment in these areas. There are a lot of numbers that are thrown around. A couple of years ago, there were 180 billion by 2030, which I think is quite an overestimate. But more recent ones, for example, this one in the CAR T therapy that just came out from Roots Analysis, talks about a 25 billion in the T-cell immunotherapy market, about in 2021 in about 10 to 12 billion in the stem cell therapy market. Just the manufacturing market could be several $billion in the next 10 years or so. So there's a lot of economic and industrial interest in this space. And there could also be a major workforce impact. Obviously, these are fairly high-paying manufacturing jobs. If you look at the Advanced Manufacturing Partnership report that the Obama administration did in 2012, that report identified biomanufacturing as a key investment area and sort of asked to partner with industry, academia, local and regional governments, as well as community colleges to build together a manufacturing workforce. So collectively, the promise and impact of cell therapies are extremely clear. The question is whether we can fulfill that promise as a nation and as a community. And I would argue that we can only do that if we as an engineering community and the clinical community and the biology community all come together and overcome the challenge of manufacturing and reproducible manufacturing. And I will emphasize the word high quality, and I'll come back again and again throughout this talk into that. Obviously large scale and with low cost, but we have to and must do it within the regulatory reimbursement, very, very critical, and policy framework. And that's not an easy task to do. We have learned a lot from the gene therapy field. We have learned a lot from the tissue engineering field and its obstacles and decade-long challenges. And I think a lot of that knowledge can be imparted here early on. So the fundamental challenge, as you all know, is that we are trying to manufacture or expand, produce, supply, distribute something that is a living entity. And even the most manufacturing process like monoclonal antibodies, that's not the case, right? Yes, we work with CHO cells, but that's not the product. The product is an extremely well-defined protein. In the virus manufacturing area, to some extent, it is true, but still the cells are so much more complicated than even that. So I give this example to a lot of my engineering colleagues that this is an incredibly complex. If you think about the amount of secretome, the number of surface proteins that are being produced, the number of microRNAs that are being secreted, the exosomes that are being secreted, we probably have about 1,000 or more biologics within a cell that could be a therapeutic. So every time we inject a cell population, we are probably injecting 1,000 to 2,000 drugs that pharma will call a drug. At the same time, and we are telling the patient we have no idea how long they will last, what the pharm PK/PD of each of those are, or how they interact with each other, not only among themselves, but among your body. So it's an incredibly powerful product, but it's an incredibly complex product. The raw materials, i.e. the source cells, are different for every patient or donor that we generate, and that affects its safety and product function. Every user is different, and the patient, the host, is an incredible variable that affects the cells directly, and we know that from various studies. And it's also a rapidly perishable product with a very complex, scattered, and a nascent supply chain, which is could be a manufacturing nightmare. So I don't want you all know this, that it's an incredibly, so far, potentially expensive therapy. There are a lot of pharma, this is sort of the Novartis story and how they are refocusing on manufacturing. This is a great article by Peter Zandstruck, came out last year, talking about quality by design principles that Isabel was just mentioning as well. And then even the Vatican is interested, and I'll talk to you about in a second. I was in a meeting organized by the Vatican on cell therapies, but the focus there is accessibility. If you're going to make $500,000 a pop therapies, who is going to have that, and whether it's going to be only for a few elite folks to have these therapies, or is it widely distributed. So a couple of years back, about 3 years back, the National Institute for Standards and Technologies asked us to put together a roadmap of challenges and barriers and what we should do in the next sort of 10 years in terms of technology development. So we came together to form the National Cell Manufacturing Consortium. This is the website. It's a fluid organization. We always seek new members, and we hope that you all who are not currently partnered would join us. But we had sort of put together about 30 industry partners. The FDA came to all the workshops, DoD, NIH, NSF, and NIST, along with 16 universities, most of them being clinical GMP centers and manufacturing centers, folks like Isabel, Bruce Levine, Adrian G., and many other folks around the country, and have an industry, clinician, academia, government partnerships to identify barriers, challenges, and talk about attaining a roadmap. And there is an executive committee of that, and we are having our sort of roadmap follow-up meeting this Tuesday in Atlanta with about, again, 65 or 70 partners coming over. This is how the roadmap looks like. I'm not going to go into excruciating details because it's freely available through the website, cellmanufacturingusa.org, but also through the government's website, themanufacturing.gov. It was officially released by the White House. This is not 13, sorry, it's 16, this past year. And it goes into very much detail, talks about autologous, allogeneic, pluripotent cells, cross-cutting technologies in a short-term, mid-term, long-term barrier space. And it has some very interesting points. Obviously, scalable differentiation process, I don't have to tell that, but integrating sensors to non-destructively gather and transmit data, QBDs, low-cost, high-efficiency genetic modification technologies, and so on, but also talks about standards and workforces. And I'll go over a little bit more details. But if I have to summarize the roadmap very early on in my talk, this is how this will look like, and the technology side of it. So some of the big challenges is lack of standards, and I'll talk about what we are doing in this space, and there is actually national efforts happening in the standard space quite a bit. Obviously, critical quality attributes. For those who are from the biopharma industry, this is bread and butter to almost any biomanufacturing, but it exists at very minimal level in the cell therapy side of things. Obviously, it varies with cell types. We don't really know what cells are the most potent and most safe. How do we measure these CQAs, and preferably in line during manufacturing, and identify batch failures very early on rather than waiting a week, 2 weeks, sometimes a month for a GMP process to end? How does quality vary with age, sex, ethnicity? We haven't even gotten there. Obviously, there is a large role of sensors and automation in this process to sort of get into the PAT, or the process analytics technologies process. Obviously, this question is a very scientific question. How do we identify the most potent cells, and how do we develop processes that only amplify those most potent cells? So scalability through quality, supply chain and logistics with built-in redundancy, that doesn't exist right now. But every manufacturing industry that the United States has led, or the world has led, it's an incredible critical component is the supply chain logistics. Obviously, transportation and distribution, and can we transform GMP with quality by design into a low-cost, low-footprint process? And the lack of trained cell manufacturing workforce. When we first met as a consortium with about 30 industry partners, one of the major things they told us is focus on the workforce, because we cannot find GMP-trained folks very, very easily, and I'll talk to you about a little more on that. I want to focus a couple of slides on the CQAs. This is a paper from the FDA a couple of years back from Steve Bauer's group at the FDA, talking about very basics, MSCs, how different MSC batches with exactly the same surface marker characteristics. So by all means, they are all MSCs. They look by current quality standards, all the greatest cells in the world, but they have completely different potency measurements. So our classical surface marker-based potency or quality measurements almost doesn't work, and I don't really know of any cells where that works. There was a great talk from UPenn yesterday on the CAR-T session talking about a deep dive into why these CAR-Ts, the CD19 CAR-Ts, were working fantastically in ALL but doesn't work in CLL as well. And coming up with a subpopulation of cells that could be a much sort of comprehensive marker. But to me, pretty much the manufacturing process has to be completely focused on quality assurance and ensuring quality assurance and functional quality assurance. So reproducibility, that ensures reproducibility, that ensures batch-to-batch consistency, and eventually, in my mind, that will get to the cost issue. If we can reduce batch failures, if we can predict quality very early on in a batch, we know we are making consistent products that's likely to success in clinical trials, then cost goes down. And how, obviously there are many ways to approach sort of the biomarker CQA issue, and very honestly, there's not going to be ever universal biomarkers. It depends upon the patient, the treatment the patient has, the disease stage that they have, but also where the cells came from, how we are manufacturing it. So, but what we intend to do at the end is generate these quality discriminators or descriptors that is likely to be multivariate. But to me, this is an incredibly integrated process of collaboration between data scientists, biologists, engineers, and clinical scientists. And you need to get clinical trial data, preclinical data, in vitro data, do large-scale either omics or other characterizations, could be biophysical parameters, could be imaging, and generate this multi-platform data analytics tools and classifiers, whether it's simple principal component analysis, whether it's some sort of nonlinear discriminators. And there is a large role of big data and informatics in this process, because without defining quality, which for a single molecule or a single protein is fairly easy to define probably, but for a complex cells is quite hard, I do not think we will make leaps and bounds progress in the cell therapy space. And if you look at this paper, again from the FDA, that just came out about a month back in PNAS, and probably one of the first papers that I have seen looking at morphological and imaging-based, sort of deep imaging-based analysis to predict MSC potency and immunomodulatory potency of MSCs. So the FDA is very much thinking in this space. They critically want to understand what quality means and how, when company A comes and says this is the greatest quality cell, to compare to company B's same MSCs that are still greatest quality cells, and figure out a way. So we as a community, as soon as we start thinking in this space, I think it's better off. Obviously, new technologies are critically needed to integrate the CQA measurements eventually with the process. And these are directly from the roadmap, what industry told us. And the way the roadmap worked, where there were no talks, no seminars, nothing, it's sort of small groups, brainstorming, prioritizing, and putting together a list of needs. I'll not read through this. You can read through it, but I'll highlight a few things. Obviously, standards has been a big thing. High-throughput assays and surrogates for cell consistency. Rapid high-throughput assays, integrated in-process sensors. And if you look at what sensors we have now in the market, it's glucose and oxygen and pH. None of them measures the product. All of them measures the process. And we got to get into sensors that measures the product and the product potency and the product function and integrate that to an in-process system. If you look at the bioprocessing, the scale-up process, media is a big challenge, and there are a lot of national efforts going on, collaborative efforts to develop universal media or consistent media. Bioreactors that have integrated information technology systems. Data is a big thing, and I'll talk a little bit about that. Feedback control. We are investing a lot in automation as an industry, as an academic partnerships, but we are rarely thinking of any of those as a feedback-controlled process, and I'll get into that in a second a little bit more. Preservation, distribution, handling, some of the key areas in this process is, and this is going to be hard and will take a long time because it hasn't happened in decades yet, is alternative to cryopreservation. But I think there are smart ideas around where you could think of sort of live cell transportation within very, very minimal bioreactor modules with integrated sensors, and there are already work going on in that process. And that could open up a whole new directions and business models, for that matter, for industry to think about. But for now, we are stuck to either cryopreservation or bedside manufacturing, and we need to make the best of that. Data and data tracking. This was repeatedly brought up by industry, especially when you are bringing in autologous samples in a large-scale regional manufacturing or a larger manufacturing setting, because a mistake in that process could equate to death, and that is a high risk. So chain of custody recording and throughout the process flow data monitoring is critically important. Standardization and regulations, and there is a lot of activities led by NIST through ISO and ASTM that are already happening, and I'll mention a few as I go along. Obviously, establishing reference materials, standards at residuals and impurities and mycoplasma, information technology standards for tracking and supply chain components, all of these are critical. And these are all laid out in that roadmap, so I really do not want to belabor those points. But one of the key things, again, I want to come back to the workforce development side, not only on the university training side, which we as academics do, I think, fairly well, but not so well in the manufacturing side. If you look at our training programs, even in bioengineering or chemical engineering or even in cell biology or immunology, it rarely talks ever about manufacturing or GMP or quality assurance or any of these. So we need to do a better job there. But majority of the manufacturing workforce in US or around the country does not come from bachelor's degree holders or master's degree holders or PhDs. They come from the 2-year community colleges, and that's the 24-hour shift manufacturing workforce. And yet there are very, very, very few community college programs around this country that even have a biomanufacturing program. There are 3 in Georgia, 3. And among probably, 50 or 100 community colleges that are out there. So we are partnering very closely with the technical college systems, and not only in Georgia, but in Massachusetts, in Wisconsin, in California, to build how do we bring in what the industry needs. And it has to be driven by industry, because we academics rarely know what needs to be done in that space. And learning from industry, figuring out what their needs and translating that. And that's a national effort that's happening, and I'll talk about that. But ultimately, we got to believe even in standardization of training. So someone trained at the Georgia technical colleges versus someone trained at the California technical colleges have a same knowledge base. If someone is a chemical engineer, I exactly know what their skill set is most of the time. But if industry doesn't know what the skill set is and how to compare skill sets, it becomes very difficult to translate. So I'll put this slide early on. I will end with this slide. This is something that needs all decks on hand, and really critically all decks on hands. Not only clinicians and cell biologists who have been absolutely critical to drive the field, but bringing in engineers from all gamuts of the space, computer scientists, big data analytics, but really bring in the patient advocates, the insurance reimbursement industry, workforce experts, industry standards organization and regulatory organization. And that's part of the goal of the Cell Manufacturing Consortium and all other national efforts. There is actually quite a bit happening in the US, and it took us probably about 3 years to get here, but in the last 6 months or so, a lot has happened. Especially the 2 very large manufacturing innovation institutes that was funded at the very last days of the Obama administration as part of their 13 manufacturing innovation institutes funded over the last administration. NIST awarded a $70 million grant matched almost 2-to-1 by industry and state government, so a $200 million institute entirely focused on biomanufacturing called the National Institute for Innovations in Manufacturing of Biologics, headquartered at Delaware, with major partners being MIT, Georgia Tech, and NC State, and probably about 30 or 40 industry partners. And then the DoD awarded an institute called ARMY with Dean Kamen from DECA as the lead, and it's called the Advanced Regenerative Medicine Institute, focusing on tissue biofabrication with a large emphasis on stem cell manufacturing at the front end of it. And again, that's also about a $200 million institute with a $80 million cash infusion from the DoD. These are 5-year institutes entirely focused to help industry. That's their primary mandate. Industry, who are members of that, sits on the executive board, decides what area the institutes will work on, floats RFP to partner with academics and other small companies and small businesses, and deliverables and milestone-driven that are yearly and 6-month benchmarks that are done. They are not for basic. This TRL 4-7 technology readiness level means that these are already bench-proof, even maybe phase 1 trial-proof, and then bring that to the market, and that's the goal of those. We have been working or proposed something to NSF. We do not know what the results of that is. That focuses more on the early new development stages, again in the cell manufacturing space. And if we get that, that's about a $40 million investment from the NSF over a 10-year period. DoD recently did a smaller initiative called MTech. There is a large focus here on the media side that I was talking about, and there is a significant industry academic consortium that's working on the media development side through that, and that's about a $20 million investment that they made. We are also working very closely to build an international partnership coalition. So obviously the National Academies recently established a forum on regenerative medicine. Cynthia is a part of that, but the FDA sits on that, industry sits on that, academics, but more importantly, patient advocates sits on that to discuss where we need to go from here. There is also a newly established standards coordinating body, and again, an industry partnership that came out of ARM, the Alliance for Regenerative Medicine. But NIST, they have an MOU with NIST, and there is a large-scale activity happening in the standards. There are at least, I would say, 3 activities happening in ISO, one on cell counting, one on cell characterization, and one on mycoplasma testing that ISO is working on right now for the cell manufacturing space. And as you know, ISO is an international organization, so there is representatives from UK, Japan, Korea, Europe, all of that, and Japan's proposing some of the other issues as well, so there is a lot of critical activities, and this is a great time to get involved, to provide your inputs, to see what you want to see in those standards, and to help ISO and NIST in that process. I talked about the Vatican meeting, and it was an amazing meeting talking about access, and the Pope gave a very impassioned talk about, and they don't have any problems with cell therapies or stem cell therapies as long as it's accessible to the common people. And so the U.S. National Center, this is something we have been trying to establish starting at Georgia Tech through philanthropic money first. So last year we established, and I'll talk very briefly about it, this is called the Marcus Center for Therapeutic Cell Manufacturing. Bernie Marcus, who is the founder of Home Depot, based in Atlanta, co-founder of Home Depot, funds actually a lot of clinical trials around the country. And he is this 86, 87-year-old gentleman who is tremendously knowledgeable about stem cells. Go figure. But the first time I met with him, he shook my hand and said, "Krish, I have better quality control for the hammers I make for Home Depot than you have for the cells you're putting inside me." Okay. But eventually he said that I'm willing, I never fund engineers, but I'm willing to fund something if you try to build a coalition with clinicians and you build a coalition with industry, and that's where we are hoping to go. So we have about a $23 million initial cash flow in there. We are building a sandbox. So one thing industry told us, we want a sandbox. We want somewhere that's really not a true GMP facility. It has every bells and whistles, but we are not producing clinical cells in there. But we want to come and test our alpha processes, our beta processes, to test our ideas in a true GMP setting. And this should be done in about the next 4 or 5 months at Georgia Tech. And we hope to build collaborations in that space. And one of the features, looking forward in that inline continuous analytic measurements, which is very hard to do in the current GMP architectural setting even, unless you put every analytical instrument in every GMP rooms. We wanted an analytical suite that can essentially sample from bioreactors in real time. And that's where we are going with that process. So this is sort of the current network that it looks like. Not all of those are done, but we are trying to put together a very robust network of people and partners, and we are always looking for new partners to work with. And this is just academic partners, and the consortium is on sort of places on top of that. What we are working on very, very briefly, we are embarking on a very large, some might call fishing expedition, we call it sort of big data approach on identification of CQAs. We are partnering with our clinical partners to get clinical sample cells, have put together a fairly large infrastructure, do pretty much everything that one can think of doing to these cells, or measuring, and then looking at potency assays, getting back clinical trial data, and you will hear a little more. There are some efforts going on to get a large clinical trial data network and database network to help the larger community. Because again, our N's are very small. If you want informatics data to be robust, you need a lot of N's. And that's a continuing process. I was at a talk, I was giving this talk at a manufacturing seminar with the CEO of Intel as the keynote speaker, and they, even to this days, have every process parameters fed back to their computational model of the process every day, every minute, and they continuously update the model based on that. And so we are way far away from that, and may not be necessary in the biomanufacturing space. But we got to think about that process, that we continuously collect the clinical trial data, feed it back to this central model of predictability, and more clinical trial data that we can input there, better and robust that models will get. But it just doesn't have to be clinical trial data. It could be preclinical data. It could be organoid models. It could be in vitro data. The other areas, we are working a lot on image-based sensors as well as measuring, inline measurement of sensors, how to automate these things. I think we are investing tremendously on organ-on-a-chip models, especially patient-derived organ-on-a-chip models, which I think is a big, it could be a critical part of potency measurements and safety measurements in a humanized physical model, not only for small molecules that I think pharma is already doing, but also for cells. Things like tumor on a chips and lung on a chips, and we are looking on neurodevelopment on a chips using iPSC-derived neurons, and so on. And then automation. And there is a lot of activity in industry right now in automation, especially in the cell therapy, especially on small benchtop end-to-end cell manufacturing platforms. But none of them really thinks about QBD or measuring CQAs or having any feedback. If I take T cells from any one of us, even as normal, healthy people, all of those source cells are different. Yet we are still culturing them for exactly the same number of days in the exact same number of cytokine milieu and ending up with 100 different products. Do we need to do that, or how that measurement of the cell source brings into the changing of the process? So automation must be flexible. And that's and a feedback control closed loop process. So flexible automation is where we need to go in the next sort of 10 years process. Ultimately, though, I get answered, how many sensors do you want to put in? How many automation? It's going to be incredibly expensive. It's sort of that development process. Once we have figured what quality means, the process takes care of the quality, and that's the principle of QBD, quality by design, that you come up with a process that ensures consistent quality, and you measure only a few things to ensure quality assurance. I'll not go into this, but I want to point into the data-driven manufacturing part for a second. Continuous feedback of process parameters into models. So there's a big role of data. Integration of data from end to end, and not only how we collect it from the cells, the patient's information, the patient's physiological and disease information, into when it gets back to the patient, and how do we integrate data. It's a huge data problem if you think you're going to manufacture it for tens of thousands of people, and that needs to be thought about fairly early on. So to me, this is a classic chemical engineer's process workflow for cell manufacturing, and you could think of many of these. But the vision that we have put together within the consortium and within the Georgia Tech Marcus Center is that we want obviously closed system manufacturing for this process. And then this could be mostly open, but we got to think about formulation and filling, how we transport, how we release test, and all of these things. But there are many, many technology development space here, and I touched upon, you think of CQA identification, both at the source and at the end. You talk about things that can real-time sample bioreactors, that can separate label-free, non-destructively potent cells, things that can do high-throughput gene editing at a very high efficiency, potency models using organ on a chips, or looking at other biophysical parameters like how these cells transport in a physiological model, looking at scaling up, transportation of live cells, process modeling. So there are a tremendous amount of engineering and engineering clinical biologic collaborative input that can enable a lot of these things. So this is my own vision. Take it with a grain of salt. What we will see in 10 years from now, robust, scalable, feedback-controlled cell manufacturing platforms that are reproducible and high quality, uniquely trained and diverse workforce. I cannot overemphasize that. Without that, there will not be a cell manufacturing industry. Affordable curative therapies, I would say millions, but that may be, who knows, maybe at least tens of thousands of patients to reach to. And enabling technologies would be these closed platforms that has feedback control and quality control in that space. And I think it's distributed manufacturing. It could be point-of-care manufacturing. That's up to industry to figure out what works best for them. But I think we got to, regardless of what that model is, I don't think the basic platform or the basic concept changes at all. It's really all hands on deck. And I'll end with sort of leave that slide up there and end my talk. Thank you.
Thank you, Chris, for this beautiful talk. This paper is open for discussion now.
Yeah, thanks for a nice talk. Just one thing. I mean, the state of maturity in the cell therapy space is not really quite there yet, and everybody's developing something different: T cells, NK cells, stem cells, et cetera. So what's your vision for standardization in light of the very many different things right now?
Yeah, so we have this discussion with NIST a lot, and NIST obviously is tasked from a government standpoint in developing industry standards. And I think the consensus here is there are probably pre-competitive basic measurement tools that can be standardized. So, the first thing they took on is we don't even agree how to count cells, right? Every one of us counts cells differently, and we'll come up with a different answer. So the first task they took in the last 6 or 8 months is standardizing counting cells. Not to tell us this is the instrument you got to use, but this is how you measure and assure or figure out how your measurement is reproducible or robust. They are working, I think Britain, actually the UK Standards Agency have put forward a document on PCR and how do we do PCR in a standardized manner. There is a couple of on mycoplasma testing that are going on. So I think there are sort of tools that are cross-platform. That's where we have to start, because the deeper standards are going to be cell-specific, application-specific, and maybe there we will not have standards, but what we'll have are best practices. And so that's hopefully is where we're going.
Thank you, Dr. Roy, for a very good overview. Let me preface my question a little bit about, about myself and why I'm asking this question. I'm a drug developer. I've been in the industry for almost 2 decades now. Mm-hmm. I've developed small molecules, large molecules, mostly in oncology. Most recently, I've been involved in this cell-based piece. In fact, I'm in this conference for the first time to learn about cell-based therapies. And my concerns have been compounded and confirmed in this meeting. There are lots of companies doing lots of new things, and the competitive juices are clear. Everybody has the best product. However, the big elephants in the room have not been discussed. For instance, cost, manufacturing, access, in fact, and you've tried to touch upon it on the manufacturing side a little bit. To me, it seems like we are following the same paradigm as we have done before. Mm-hmm. My question to you is, and you did this in the second last slide, what are the paradigm shifting out-of-box ideas that we need to think about? Do we need to have, instead of pharmacies in hospitals, cell-based manufacturing units created within hospitals? I'm asking clearly out-of-box ideas. Do we need to put in manufacturing courses for all PhD students? So that's what I'm looking for, instead of the same old thing that we hear in every conference.
Right, and this is exactly the purpose for this. So, for sort of the 3 years or so, we asked that this is not going to be an entirely industry-driven effort, because they either don't have the time or the resources to spend on that front-end stuff. It has to be a federal-driven effort, and the argument there is long-term benefit to the economy if you can make it successful. All of that above. So there is a lot of effort in industry going on benchtop, point-of-care, bedside manufacturing. But again, it's just automating a certain process that does not change from patient to patient or anything. There is really no quality-driven, feedback-controlled automation process yet that at least I know of. There is a large debate in industry whether you do regional manufacturing. I think most would agree that the centralized manufacturing concept that pharma has is probably may not work unless we have a completely allogeneic process. So regional manufacturing spaces, distributed, and point of care, I think both will happen. And both are happening. It's not that. As far as the workforce side, yes, I think we have done a very poor job, even for the current biopharma industry, in integrating manufacturing concepts in our curriculum from anywhere from technical colleges to PhDs. And I think even retraining, how do we retrain the current biopharma workforce to work in the cell side? And that's a logistical challenge. But the nimble and the Army that we talked about, that large infusion of funds that DoD and NIST has given, about 40% of those $200 million each would go towards workforce development. And I think it behooves the industry to take part in that process if they see that to be one of their bottlenecks.
Thank you very much, Chris, for this very stimulating discussion. I think job security is certainly up to date.
So when we were organizing the session, we wanted to actually invite some case studies, so some of the pioneers in manufacturing for the emergent engineered cell therapies. So the next speaker will be representing Strombelis from GSK, so Michelle Myers, and this was an approved product from last year, so hopefully Michelle can apply some of her personal experiences with the challenges that Krishendu just presented.
There have been a lot of personal experiences and challenges, that's for sure. So yes. Thank you for inviting me. Thank you for coming out on a Saturday morning, the last soggy Saturday morning of the conference. There are a lot of lessons learned from what we did for Strombelis, and we're still learning our lessons as we move on here. What I want to talk about, I really don't need to show, we're focused on ex vivo gene therapy right now at GSK, and I really don't need to show this picture to this audience, but I show it to point out one important point here, and that is that essentially for all these products, we don't have just one process to manage. We've got to manage, got to manage the cell process, and we've got to manage this virus vector production. So essentially, we have 2 control strategies to put in place. We have 2 process qualifications to do. We have 2 comparability strategies to put in place. We have double everything. There's 2 module 3s that have to be prepared. And so that's an underlying piece here that goes with everything that we're doing, and why I think, yesterday when Dr. Maria Grazia Roncarlo was showing her timeline, it struck me from 2010 to 2015, from the time GSK got involved until the time that we got the file in. Why did it take us so long? I want to share some of those stories, but the underlying piece is it's double. It took double, and we started with 5 people in GSK working on this program. So those are part of the challenges that go along here. GSK has gotten involved with cell and gene therapy through our collaboration with the Telethon Institute of Gene Therapy. This was driven by GSK's commitment in rare diseases back in 2010, with the idea that Telethon is responsible for advancing all of the programs to proof of concept, and then GSK is responsible for driving it into the market, the global regulatory manufacturing and all that commercial activity. So get your arms around the control strategy, get your arms around comparability, process qualification, put the file in place. We started with a program for ex vivo gene therapy for ADA SCID, which is Strimvelis. And I'm happy to say that we filed it in 2015. Last year, we received approval, and just last month, we treated our first commercial patient. So it's been a long journey, but I'm pleased to have had the opportunity to participate in. And we're really excited about the follow-on therapies. First, for metachromatic leukodystrophy and for Wiskott-Aldrich syndrome. Now, GSK has bigger plans for other programs in larger patient populations and larger indications, and those are a whole separate bunch of challenges that we're working on to try to scale it up beyond these ultra-rare diseases. But what I want to focus on here today are those things that we're doing for these ultra-rare diseases to get these programs into the commercial setting. It's important to point out that all 3 of these programs are ultra-rare diseases. All 3 of these programs have shown wonderful efficacy data in the clinical trial, and GSK has gotten involved in them after that proof of concept. So for all of these therapies, CMC is rate limiting. We are the ones holding things up from getting these into the market. If anybody's ever been on critical path before, you know the heat. But what I want to spend the rest of the talk on is talking about, again, what we've done for Strimvelis, what we're still doing for Strimvelis, and what we're doing for these other programs. So one of the first things we had to do was get our arms around all of the data, the historical data, the manufacturing data, the clinical data, and put a control strategy in place. And everybody says, well, okay, control strategy means so many different things to so many people. So I want to take a little bit of time and walk through exactly what we mean by control strategy. So to do that, we go through a series of assessments, asking a series of questions. First, what are the critical quality attributes? What is important to safety and efficacy, just as Chris has pointed out? And then linking the parameters in the process. What are the critical parameters? How are the parameters linked to those quality attributes? How are the raw materials linked to those quality attributes? What are those critical raw materials? And then finally, what is the testing strategy? Based on the controls in place, how are we going to test this product? So let's start with the critical quality attributes, because that's really the foundation of everything we talk about in terms of control strategy. What are critical quality attributes? The ICH guideline definition up there. Importantly, what we see are that it's the attributes of the product that are important for safety and efficacy. It's the baseline for all the rest of the control strategy and used many places in what we're doing to manage the comparability and so on. Importantly, what it's not. CQAs are not analytical methods. Flow cytometry is not a critical quality attribute. The immunophenotype of the product certainly is, but flow cytometry isn't. It's not a specification. Just because we've called something a critical quality attribute doesn't mean we're going to always make it a release test. So I think those are important distinctions to be made. And you would think that especially for an autologous therapy where you have the clinical outcomes and you can link it right back to the batch of the product, you'd think that looking at each critical quality attribute in terms of its safety and efficacy would be relatively easy, but that's not always the case, especially in a company like GSK where you've got to have the clinical folks talking the same language as the CMC folks and understanding the characterization of the product. So that's been a bit of a challenge for us, but there's some good work that's been done to help us understand these critical quality attributes. This is just one example. This is looking at vector copy numbers. So what's shown here is vector copy number, CD34 per kilogram, and the vector copy number, and engraftment. And while there aren't a lot of data, we do see a statistically significant trend. And so what we can do is use these data to support our assessment for each attribute. So we comb through all of the clinical data to understand what's really important to patient safety and efficacy, and put this assessment together so that we have clear rationale for what we're doing. So for ADA SCID, copy number is a critical quality attribute. We know that it's important for long-term engraftment, but if we go too high, we know it's also a safety risk. If copy number is too high, we worry about insertional mutagenesis, and so we don't want that vector copy number to go so high. So both in terms of efficacy and safety, we've rated it a very high score, which automatically makes it a critical quality attribute. As I said, we've got to do everything twice. So we have to go through it for vector. And you would think a lot of times we go from the vector process to the cell process, and everything moves in that direction. But what we found is when we sat down to first go through the vector critical quality attributes, because we had all those data, it was easier to start there, we couldn't do it, because you need the critical quality attributes for the cell product before you can do that for the vector product. So that was that was one of the lessons learned. But again, we're here, oh, sorry, oh, went too far. Here we're looking at the infectious titer and copy number. And again, the data are pretty scattered, but we do see that they're statistically significant. And so what we've identified infectious titer as another critical attribute and rated it high in impact in terms of safety and efficacy. So in terms of the vector process, we have to pay attention to that infectious titer. Now, these are just two examples that are relatively easy to imagine. You can imagine those that are at the other end of the spectrum, and you can imagine the debates that go on for those in the middle of the spectrum. Once we have the critical quality attributes, we can go back and start looking at the process data, dig through the process data that we have for the products, and understand that for ADA SCID there were 18 patients that were treated when we got all of the data. And so there was a fair amount of data, but only 18 patients at the same time. But then you have to go through those piles of batch records to pull out what's important here and understand what critical parameters and what are the raw materials that are linked to those critical quality attributes. Once we combed through the data and found what we knew to be critical, then we get into the good part. We get into how are those parameters and raw materials being controlled in the process now. So this is getting way down in the weeds. This is asking the question, what are the operators on the floor doing to ensure that those critical parameters are always, to ensure that that seating density is always what it's supposed to be, to ensure that the temperature is always what it's supposed to be. What are the control mechanisms that exist? Those underlying control mechanisms make up that larger control strategy. And that's the meat on the plate. That's the part that we've really got to get our arms around for all of these processes. Once we have that, once we know what's critical, the critical quality attributes are, and we know how well it's being controlled, we can then go in and ask, okay, what do we need to do in terms of testing, in process testing or release testing? There are some things that you can imagine, vector copy number, infectious titer, those things I just went over, pretty easy to imagine. Those are going to be on the release specifications. There's others that you can imagine, not so important, not a big deal. But again, it's those things in the middle that cause debate. And so we put a system in place, as GSK does for most everything, to use a consistent rationale for what we would include in the release testing. And that's what's shown here. So it takes into consideration the criticality of the attribute, which I showed in some of the assessments a couple slides back, and it takes into consideration how well you're controlling it. So we ask a series of questions to understand how well we're controlling, how well we understand how that attribute is being controlled. So is there a good understanding of the process and the effect on the attribute? If not, we give it a fairly high risk score in terms of its process impact. If you can, are the parameters in the raw materials that impact the quality attribute well controlled? If they're not, you're certainly going to give it a high process impact score. Are the data from process qualification, all the data from manufacturing, are they within the range that you expect them to be? If they're not, again, you got a control problem and it needs to be a high risk piece. Is there a significantly wide acceptable range for this attribute? If vector copy number can be here, okay, but if it needs to be here, then that's a different story. And are you controlling within that range? So it's asking that question and ranking the risk associated with that. So if everything looks good, it's a pretty low risk. If you've got things, you answer no to some of these questions, and you come out with a higher process risk score. So we take this critical quality attribute impact score, how important is the critical quality attribute, and we look at this process impact score based on how well the process is controlling it, and decide, is this an attribute that we don't really need to worry about in release testing, or is this an attribute that we should be testing with every batch of release? And then, of course, we have some in the middle, some that we look at as part of process monitoring, process comparability as a result of process changes. We'll come back to these as well. Of course, this is where all the debate occurs as well. But we use this system to try to get our arms around all of the data and apply a consistent rationale for what we do and ensure that we have clear definition about how we're managing our process. So with this in mind, how do we use the control strategy? First, articulating in the file, convincing one another, convincing the regulators that we know what we're doing in terms of control strategy. So having that vector copy number as the foundation, that critical quality attribute. What are the process parameters that are impacting it? What are the raw materials? And what's our testing strategy? So it's thinking about these three pillars of the control strategy and how they support the critical quality attribute. So for each critical quality attribute, we talk about its control strategy. So that's the way we're using it to communicate with one another. Another tool for communicating it is shown here. So here, looking at the inputs to the process, each step of the process, the isolation of the stem cells, their stimulation, their transduction, and then the wash and the formulation. And looking across at each critical quality attribute. For example, copy number, we know that some of the input materials have impact to them, so those are in the red category, whereas we're not so worried about copy number when we're doing the isolation process. And so on. It's understanding each step of the process. Here, for example, clonogenic potential, we know there's a lot of things in the process that impact clonogenic potential. There may be things during the last step in the process that would impact clonogenic potential. This is a way we use to communicate the control strategy again. And importantly, it's a way that we communicate it with the operators on the floor. It's been a great tool for us when deviations have occurred, when we've had things out of range. We come back to this and say, okay, if the seating density is out of range, what impact is that going to have on copy number? And we can go to that piece of data and pull it out. So it gives us a way of organizing it, especially in an organization like GSK, where we're not just trying to, we're trying to educate one another, we're trying to get the rest of GSK, including quality and regulatory and all of our partners, up to speed with what it means to put these products on the market. This has always also been a great tool for us in terms of technology transfer, which I'll talk about in a little bit. But understand that all of this was happening for Strimvelis. All of this was happening in an iterative process while we were also considering a number of process changes and trying to work through comparability. So control strategy, everything was happening in parallel as we were doing some development work to look at process changes. But we got all of the data in and we said, okay, well, why do you need to make any changes? This product works. You need to get it on the market. Just do it. Don't make any changes. What do you need to do? It's a low volume product. There's an acute need. Just get it out. But when we looked at all the data, we saw some variability that caused concerns, some concerns about process robustness, raw materials. There were still some animal-derived raw materials in there, like porcine trypsin we wanted to move away from. We wanted to decrease residual impurities, include some more wash steps, and containment. This drove our aseptic guys crazy. You've got a company like GSK that's used to making sterile parenterals, and you tell them you're going to make a product in a hood, you're going to go in and out of the hood a few times, and oh, by the way, you have 6 hours to release this, so you're not going to do sterility testing. I mean, it was tough. There's a lot of smoking going on with it. So we can't do this. So there was a lot of work done to try to close the process up and put some more aseptic controls in place. So we had to do something, but not everything. We had to generate development data and make changes introduced with an aim to increase the scale of the manufacture for the vector, increase the process efficiency, improve process performance, robustness, decrease the variability in the process, and of course, increase the sterility assurance. But we had all the clinical data. You can't go change in everything so that it could potentially require additional clinical data. And oh, by the way, patients are waiting. Get this out. Get this to the market. So one of the things that we move forward with then is to input those changes, and then how do we assess, well, what do we do for comparability, and how do we assess the impact of those changes? Again, we went back to the critical quality attributes and asked the questions, what impact were we having on those critical quality attributes? Starting with the vector process, we made some changes to the vector process, knowing that we were going to impact infectious titer, infectivity. So those were certainly areas we had to focus on. And they would have the knock-on effect of vector copy number, transduction efficiency potentially being impacted. And then as we were closing up the cell process, there were potential impacts to other stem cell potential, cell viability that we had to take into consideration. There were a number of things we were able to say, no big deal. We're not impacting safety. If anything, we're going to improve safety. We're going to include some wash steps so that process-related impurities would go down. So those were things we felt pretty confident in, still had to characterize them, but felt pretty comfortable about it. And so this is the comparability strategy. Of course, again, 2 comparability programs, 2 comparability studies. First with the vector, comparing the clinical and the commercial vector processes. Importantly, completing stability and in vitro comparability for the vector from the commercial process and the clinical process. And again, going back to the assessment that I just showed you for identification of those attributes that we had to include as part of that characterization. Study 2 then was using that clinical or commercial vector in a clinical and commercial cell process. So starting with a common starting material and breaking it into 2 arms, one in the clinical arm and one in the commercial arm, and then looking at the stability and the characterization comparability data associated with that cell process, going through the clinical and commercial process with the clinical and commercial vector. And I'll show you just one piece of data out of this. So this is looking at the clonogenic potential for the clinical arm of the process and the commercial arm of the process. And fortunately, what we see is no difference between those two arms, which is nice. We see that this is where the development data were. So as we were making those process changes, we saw that the development data, the clonogenic potential, was also within that same kind of range. What you see down here is the min and max of what it was from the clinical data. So there's a significant difference between the clinical data and what we saw here in our studies, and we attribute that to the fact that we have to use healthy donor material in all of these studies. So healthy donor material having a different clonogenic potential than what we saw for the patient population. Fortunately, what we could do then is look at the change in clonogenic potential. So from the point at which the CD34 positive cells are isolated to the drug substance, we could go back and say, okay, did the process changes that we make have any impact on the clonogenic potential of the cells between those 2 points? So we looked at that delta. And again, Again, what we see, the clinical and the commercial arms were very much the same. Here are the development data, and here are that's the delta observed for batches in the clinic. So we didn't see any difference between what was happening in our improved process and what was happening in the clinical process. So I show this data to illustrate the complexity that's involved here in setting acceptance criteria for these comparability study. So you have to take into consideration these two arms of the study. We had to take into consideration the development data that we were able to generate, and we had to take into consider the clinical data that we had as well. So, and interpret all of those data in the context of the patient population, the healthy donors, as well as any changes to the analytical methods. Not that that ever happens, it's all very easy. So that's all part of what we had to manage to move Strimvelis from the time we got involved in late 2010 until when we got the file in 2015. This is the work that was going on. These are the changes that were being put into place. This is all the complexity that we were trying to manage, get written up, compiled, and make in a decent story for regulators could follow. But of course, we can't leave it there. So post-approval, we are looking at some changes to try to expand the supply chain. So right now, for Strimvelis, the only starting material that's used is bone marrow. We want to move to a process where we could use either bone marrow or mobilized peripheral blood in order to treat larger patient population and potentially older patients. Right now, the final product is fresh, and it's fairly easy to do, fresh product within 6-hour shelf life. The product is produced at Molmed, our CMO in Milan, and then it's walked across to the Hospital San Rafael where it's administered. So that's a relatively easy way to manage the supply chain now, but that's not sustainable, and that's certainly not something that's going to get anywhere close to the patient access that we'd really like to have. So that's why we're talking about cryopreserved product and looking very hard at how to manage cryopreserved product. So what do you need to do to get cryopreserved product out there? These are just some of the development things that we're doing, certainly developing the formulation, developing the control rate freeze conditions, determining the stability pre and post cryo, figuring out how we're going to thaw it. Once it's thawed, how are we going to get it to the patients? What's the time that we have to get it to the patients? Shipping validation, temperature excursions, all of that. We're generating those data now to study the impact on product quality. Of course, all of those data help us in our impact assessment about the CQAs impacted by the change. And that's just shown here. Some things are pretty easy. Microbiological control residuals, low impact. We don't expect anything to change as a result of cryopreservation. Of course, having the ability to have all of the testing done is a huge benefit for both us in terms of managing the product supply, as well as the patients, because currently the patients are starting that immunoablative therapy while the product is still in production. And that causes a fair amount of agita when you're in production as well. And God forbid anything should happen to those cells. God forbid any contamination were to happen. Other things with cryopreservation that we're concerned about, copy number. We're looking closely at the impact on copy number, and certainly the percent CD34 positive, the stem cell potential, and the cell viability are all things that we put in that high-risk category. So we're generating data, trying to understand the impact on these critical quality attributes, and then using that to design the comparability strategy. So again, the comparability strategy, starting from the healthy donor material, going through the process, and then looking at the two arms, the fresh and the frozen, from the bone marrow process, and then for the mobilized peripheral blood, running through that process and looking at that frozen product. So the question comes here, how to set these acceptance criteria, what to expect, and how to manage the acceptance criteria between bone marrow and mobilized peripheral blood. We know there's going to be some differences. Are those differences important? What impact do those differences have? How do we manage those differences? We certainly want to have one drug product from either source. I think that's possible based on our critical quality attributes. We need to look at the subpopulations of CD34 positive cells as well. One of the things we're learning as we go through this is the operational challenges involved with doing these kinds of studies. We have to ensure that we have a sufficient amount of material in that healthy donor material to start with in order to have a sufficient amount of drug product and a sufficient number of cells for all the characterization testing we want to do, as well as the stability testing that we want to do. That's a big challenge. To put the cells into the final container closure at the concentration of the target cell concentration for the final product, you need to start with a heck of a lot of bone marrow to get enough cells to do that. The other thing is having enough resources to do all the arms of the study at the same time, to run things in parallel, to have a fresh and a frozen, to have all of that going on at the same time. We have to make sure we have the resources available to do that. So, but that's the work that we're doing with Strimbelis post-approval. The other programs I talked about, the MLD and Wiskott-Aldrich syndrome, what we're doing there. Right now, ADA-SCID, for the treatment for ADA-SCID, Strimbelis has only been approved in Europe for MLD and WAS. We're wanting to gain licensure both in Europe and in the US. So we've started tech transfer. We've started tech transfer for a product that's only been made less than 50 times in Milan by a small group of people. So you can imagine the challenges there. In addition to that, we are wanting to implement those process changes. So in Milan and the Hospital San Rafael now, those are fresh formulations, starting with only bone marrow. They've introduced some use of mobilized peripheral blood. We're wanting to incorporate those changes into the files for MLD and WAS as well. And in addition, we have to think about the starting material. I haven't talked a lot about the starting material because that's been pretty easy for us for Strimbelis. The starting material came out of Hospital San Rafael and came across to MolMed. They walked that across, they walked the product back. That's a pretty relatively easy supply chain. We know that that's not going to happen in the US. Nobody walks anywhere in the US anyway, right? So we've got to manage the starting material and its transport from point A to point B. And how long do we have to do that? We have to understand that input material stability, it's times, temperatures, conditions that are involved there to make sure that we start with a product, and we start with the material to get the product of appropriate quality. So what we're doing here, again, process transfers. We're working with our contractor in the US, Wuxi Aptech, in the Philadelphia Navy Yard. It started with documentation, training runs. We're working in engineering runs this summer, working to get into process qualification and comparability late this year for metachromatic leukodystrophy. It's been a bit of a challenging road, certainly some lessons learned, but we're making good progress. Comparability, a whole nother can of worms for us that we're working through. To demonstrate comparability for a process in the US and a process in Italy, and the logistics involved with how do you split that starting material, and how can we ensure that that's going to work. That's a whole new challenge for us. But to even do that comparability, we have to also move the analytical methods over, which has also been a bit of a learning experience for us. Again, starting with documentation, training, writing the transfer criteria, and then importantly, demonstrating that the analytical method is comparable to the method as it's performed in Milan. So we can't demonstrate process comparability if our analytical methods aren't comparable. So we have to start with that comparability first to make sure that we're ready for that process comparability. The analytical methods can't be underestimated. I show this as a list of what we've been doing. The methods for raw materials, compendial methods, this is supposed to be the easy box. We have had some challenges there, but we've worked through them. Product-specific methods, certainly immunophenotype, clonogenic potential. We have to think about the operators and the gating, and training operators up, ensuring that counting the cells on those plates for clonogenic potential, no small task, ensuring that they are doing it in a comparable way to operators in another continent, no small task. Lentiviral copy number, again, we're going to need to demonstrate comparability in copy number, transduction efficiency, and the transgene product expression. So that's all work that's underway right now. So just last slide, key takeaways. I hope what I've convinced you is that the control strategy as a basis for this work has helped us a great deal to make sure that we have consistent rationale and a systematic way of getting our arms around all of this data and being able to communicate it both to regulators and to operators across the spectrum. Those assessments provide us, again, that consistent way of getting the testing strategy in place. The changes in technology transfer that are required for global access create this requirement for these complex comparability studies that can't be underestimated. But the control strategy can be leveraged to help us design what's important here and focus on what's important in those comparability studies. And then finally, I don't want to sound cocky, we're certainly not that. But I think we've learned from Strimvelis, although we do have to be careful about the whole idea of saying, oh, that's just the way we did it for Strimvelis, just do it that way. We're very conscious of not doing that, and actually the amount of turnover that's happened in GSK and is helping to manage that in and of itself, by itself. But I think we're well positioned. I think we've learned, and I think we're well positioned to bring MLD and WAS to the patients who need it. And finally, some acknowledgments. There's huge groups of people now involved. As I said, when GSK first got involved in 2010, there were about 5 people committed to cell and gene therapy. The team is definitely growing. Some folks who've been there to support Strimvelis have left, but we've also had great partners at TGIT as well as at MolMed and with WuXi AppTec in our recent transfers. So thank you for your attention. Happy to take any questions.
Thank you. That was really inspiring to hear, seeing as we're starting to address some of these challenges ourselves. So we'll take questions.
Can you comment on how patient material, starting material, will feed into comparability analysis with process changes? Because you did mention that donors, healthy donors are quite different.
Yeah, they are different, but we're not using patient material for any of that comparability studies. It really wouldn't be ethical to say, oh, we're going to take your material and split it between 2 processing sites, and you might get some of the cells back. What we're trying to do, though, is to characterize it. We're trying to characterize the patient material and understand the differences between the patient material and the healthy donor material so that when we set those acceptance criteria, it all makes sense. And understanding that there are some quality attributes that we'll have to just compare those two arms, and other quality attributes where we can go back and look at those clinical data because there is less of an impact between healthy material and patient material.
So there's just not physically enough out of a patient to be able to...
No, and we really wouldn't want to play around with it, to be honest. Those are cells important to get back to the patient.
Hi. Very nice talk. My question is, you have a product, the shelf life is just 6 hours. Do you have a challenge regarding release the final product against your, release assays? Do you finish with the release assays and then, how to handle that?
We've prioritized, we've taken some of the sampling at drug substance, so we have a little bit of a window having tested the drug substance. And it's two-stage release. We're using a two-stage release strategy, primarily looking at viability of the cells, the cell concentration, and then we are also using rapid microbial methods throughout the process, so looking at use of the BACT alert system and looking at those points, the sampling at those points that we deem highest risk to understand, is this product, what are the risks to administering this product to the patients?
That's fantastic to see the real life story, right? So it took 5 years. In your experience, what would bring it down?
Well, I like to think it took 4, because
Okay,
we didn't really
sure.
get until 2010, and we had a file pretty much together by the start of 2015, but it took 4 years.
Great.
So I'm sorry, what does it
No,
take
I
to
mean,
bring it down?
yeah, so I'm sure your patients would have wanted it to be in 1 year or less,
Yeah.
right? So how do you bring it down? And then sort of the corollary to that question is, probably you could bring it down by if your clinical partners would have done a lot of these stuff or were aware that these needs to be done. How do we transfer this CQA or process robustness to labs earlier on?
Oh yeah, it's definitely a conversation we're trying to have earlier on to consider the control strategy. I think, how would we have reduced that 4 years? I think the resources as well. Again, building the team, building the capability, understanding, getting our heads around the problem statements to start with was probably the first year to just understand all of the complexity that we had in front of us. And these data were, there was a fair amount of variability in these data, so we had to try to ascertain what was important and how to manage to reduce that variability. But that variability also works to our advantage, I just want to point out, because if we found that vector copy number, that the product is efficient with vector copy number from here to here, that's a great window to drive that bus through. That's not to say that we shouldn't be aiming for a narrower target. So what would I do to try to, what are we doing? We're putting in place better, more capability, the teams, the resources to do it. We've got more, we're learning from what we did previously. I'm not sure how we could have done it differently to have reduced that time back in 2010. If I had a time machine, it probably wouldn't look a lot different. Just because we didn't know those things and we didn't know the questions to ask.
You could start with more than 5 people.
Yeah.
Okay. All right. Thank you,
Good,
Michelle.
thanks.